Okay, so the webinar is about to begin. Am I audible?
Yes.
Namaste and good evening all. Respected teachers, colleagues, dear friends, students and distinguished speaker of today's webinar, Mr. Harry Jose, Associate Vice President and Global Head of Marketing, Syngene International Limited, Bangalore. Honorable panel members, Mr. Manoj MN, Dr. Dilip KJ, respected teachers, dear students and fellow pharmacists. On behalf of COPS Global, I extend a warm welcome to all the participants who have joined us today. At the onset, on behalf of COPS Global family, I wish all my friends, teachers, and dear students present here a happy Republic Day as our nation celebrates our 73rd Republic Day. It has been more than 2 years since our life has been changed by COVID. It is a time of uncertainties for all of us, and especially for students. The fear of the pandemic on one side and their future on the other side. Their course of study is getting delayed, whether to go for a post-graduation or to opt for a job, where to go, full of uncertainties. It is here the COPS Global offers to extend their support. COPS Global is a collective of all those graduated from College of Pharmaceutical Sciences, Government Medical College, Tirunelveli. It is an initiative to help students who are left in the dark to show a light, to choose a better path towards a successful career. And one such initiative from COPS Global is this international webinar series, Pharma Fair, a guiding light for the next generation pharma professionals. Today, it is the fifth webinar in the series. The topic of today's webinar is an overview of BioPharma R&D. Today's talk is delivered by Mr. Harry Jose, Associate Vice President and Global Head of Marketing at Syngene, Bangalore. Syngene International Limited is one of the leading global contract discovery and development and manufacturing organization in the pharmaceutical and biotechnology space. Mr. Harry Jose, an alumnus of COPS Trivandrum, graduated BPharm from COPS Trivandrum in 1999 and then pursued Masters in Business Administration in Marketing from the prestigious Nirma Institute of Management, Ahmedabad. Mr. Harry Jose will be taking us through the various nuances of drug discovery, vaccine development, research and development in the pharmaceutical and biopharmaceutical industry. We are indeed fortunate to have with us today a person who is competent and having hands-on experiences in the various fields of research R&D. On behalf of all the participants here, I extend a hearty warm welcome to Mr. Harry Jose. Mr. Harry Jose, welcome.
Thank you.
In the panel today, we have Mr. Manoj MN, Research Team Lead, Big Tech Labs, Bangalore, and Dr. Dilip KJ, Professor of Pharmaceutics, College of Pharmaceutical Sciences, Government Medical College, Alappuzha. I extend a warm welcome to Mr. Manoj MN and to Dr. Dilip KJ. Welcome, sir. The target audience of this webinar series is our dear students. These webinar series are being organized especially for the students. I extend a warm welcome to all our dear students. I also extend a hearty welcome to all our teachers and those working behind the screen. Dr. Prashant SS, Mr. Thomas Paul, Dr. Soni Paulos, Mr. Shabir, and the list goes on. Welcome to this webinar series. So dear students, please make use of this opportunity. I'm sure that you will be having a lot of queries. Queries are always welcome. Please make use of the chat box. Our experts will be happy to answer those queries. Please give your valuable feedbacks. A link for the feedback and for the e-certificates will be given towards the end of the webinar. Please don't forget to fill and submit the details in that link. E-certificate will be forwarded to your mail as soon as you complete this process. Wishing you all a happy, informative session. Thank you. That's it from my side. May I invite panel members to take forward the seminar. Over to you, Mr. Manoj Aman.
Thank you, Ranjesh.
Thank you.
And welcome, everybody. And it's my pleasure to invite today's speaker, Mr. Harry Jose. And as Ranjesh mentioned, he's an alumnus of COPS Trivandrum from 96 to 2000. And he has done his MBA and he has worked in various industry as marketing personnel. Before joining Syngene, he was the head of marketing in Infosys BPM, and he has even proven his career there. And since 2018, Harry is the head of marketing and associate vice president at Syngene International. And as you all know, Syngene is a key player in research and development in South India and based in Bangalore, and they are into multiple pharma and biopharma segments. And it is good to have a speaker who has live experience in these sections, and good to listen to from a key player directly that always have a value in that. And as you know, this is our last webinar in the current series. This is the 5th webinar, and we will be starting new series next month onwards with much more divergent and new topics. And we value your feedback. And if you wish to know something in detail on a specific topic, please give that in the feedback form, and we'll try to arrange specific speakers for that. So Harry has done his BPharm and MBA, and I think he's still pursuing his academic career. He is pursuing his leadership management at the prestigious Kellogg's Institute. Harry, congratulations, and that shows his interest in this sector, and he's always floating on the forefront, and he's showing he's sharpening his skill sets. That's a good message to all the students that he's not stopping his studies, he's still pursuing. So with that, Harry, I'm not taking much of your time. Let us dive in, and it's very interesting sessions. We had a lot of requests for research and biopharma research, and I'm sure Harry will be able to give a very good overview on that. Over to you, Harry.
Thanks a lot, Manoj. Just give me a second while I set up my screen. Right. Cool. I'm audible, right? And you can see my screen?
Yes, Harry. Please
Audible
continue.
and clear.
Cool. Thanks a lot. So thanks a lot for that wonderful introduction, Manoj. Today's session is an overview of the biopharma landscape. So it's a pretty wide landscape, and it's not that easy to cover everything in the limited amount of time that we have. So I'm just going to try my best and request you to follow along, and please type in your questions in the chat window so that towards the end of the session we can take that up, right? So before we start off, a quick disclaimer. As always, the content and views expressed in this deck and presentation are mine alone, does not necessarily reflect that of my employer. I'm not a scientist. I am essentially an industry watcher by profession as well as by passion. So the content of these slides are designed to give you a very broad overview of the direction in which this particular industry is headed. For the science part, follow this up with your own research. And I have tried to give exhaustive links in practically all of the slides, and those links would be a very good starting point for this. Right? So let's just kick in. So as Manoj mentioned, I was an alumnus of COPS College in Trivandrum. So when I was doing my BPharm, practically all of my friends in the medical college men's hostel had this particular book in their rooms. This is a book by Eric Siegel. It's called Doctors. It essentially goes through the lives of students at Harvard Medical College, okay, as they proceeded through their MBBS course. Now, within that book, there was this one sentence which has really stuck with me, and it goes like this. And this is from the inaugural session given by the Dean of Harvard Medical College to the new joinees, so new students who were freshly starting their MBBS course. And it goes like this. I urge you to engrave this on the template of your memories. There are thousands of diseases in this world, but medical science only has an empirical cure for only 26 of them. The rest is guesswork. it just kind of shows the limitations of, both the medical science, the pharmaceutical industry, and all of the associated life science, industries put together. This book was published in 1988. Obviously, the science has kind of, come along quite a bit since then. And it probably, would be interesting to kind of, look at how many diseases do we have a cure, right? So, but before we proceed, I just wanted to kind of set the context by talking a little bit about drugs themselves, right? How do you select, how do you validate a drug target? To put it very simply, the ideal drug would have 4 characteristics. It selectively binds with the target. It reaches the target easily, does the job for which, we are creating it, and it is not toxic. It is not toxic to human beings. So by a target, we typically, most of the targets are proteins, right? Within those targets also, it could be, there could be ion channels, right? The enzymes. So these are the kind of, typical targets that you have. And when you look at selecting a target, there are really 7 factors that you need to consider. The first one is there an unmet medical need? That is self-explanatory. Is there a large enough patient population? So there are several rare diseases, which do not receive enough attention simply because there is not, so much of an patient population for that. Do you already have a proof of concept? Are there existing drugs in this space? That is what precedence means. How easy is it to develop the assays to test the efficacy and the various physicochemical parameters associated with a drug? Do you have availability of animal models? Now, this might be something which you might be familiar with, but this is pretty much interesting because if you have a good animal model, it is quite possible that the drug that you develop will stand the test of time. A very interesting anecdote in this is the discovery of penicillin. Of course, everybody knows that penicillin was discovered by Alexander Fleming, but it took a long time for the drug to actually reach the market, right? So when Florian Chain actually, did the initial synthesis, the animal model that they used was that of Swiss albino mice instead of the regular guinea pigs. Now, had they used guinea pigs, for reasons which we do not still know, guinea pigs do not tolerate penicillin. Had they actually gone for guinea pigs, the discovery of penicillin could have taken several more years because it would have shown up as a failure. Luckily, they went with Swiss albino mice. So that's something which actually shows how important an animal model, a proper animal model, is for identifying and discovering drugs. Again, when it comes to COVID, some of you may know that ACE2 receptors, right, which is through which the COVID virus gets into the human body, is absent in mice. So in order to test it in the mice models, literally we had to create transgenic mice by literally, putting ACE2 receptors into the mice, so that you have mice with ACE2 receptors so that you can actually observe how the drug is working, right? The next part is, are you going with biologics or small molecules? I'm going to extensively cover that in the coming up slides, so I'll just kind of speed up on that. And finally, how easy is it to synthesize the chemical matter? Right, so these are factors, and I've given a source below, Medicinal Chemistry for Practitioners by Jack Lee. This is an extremely good book, and if you get a chance to read it, I would definitely suggest that you go for it. Right, now back to those, the limited number of diseases that, we encoded in the first quotation. What we don't know today genuinely dwarfs everything that we actually do know. So there are 20,000 genes in the human body. So around, the 2010 time frame, when the Human Genome Project was completed, okay, we discovered that, there are 20,000 plus genes in the human body, but 60% of these, we really do not know what it does. We call it junk DNA or non-coding RNA. We just don't really know what actually, it does. Okay, now out of these 20,000 genes, and we know that there are over 20,000 proteins which are coded by these genes, only 700 of them can be targeted. And proteins are the number one target, for drugs, right? And there are over 10,000 diseases known to mankind today. Just about 500 of them have a treatment. And we've just released, FDA in the US has so far approved, I think, about 2,000 plus drugs. So that really shows how much more we need to go. Of course, we have come a long way in the last 100 years, but there is still a huge amount of ground to cover. And this also shows the tremendous potential associated with this field. And this really, this is the thing that you need to keep in mind when you are pursuing a career in the pharma industry, right? Moving forward, this is a graphic which hopefully should be fairly familiar to you. This actually shows how the drug discovery and development actually happens and why it is such a long and risky road. So once you have identified a target, which we said in the previous slide, we said that there are quite a few protein targets, right? So once you have identified the targets, most of the time you have to go through over 5,000 to 10,000 compounds to get a hit. A hit is something which actually shows the drug efficacy that you want. Now, then you have to go through a lot of, identifying leads from the hits, going through a lead optimization phase, all of the preclinical part, going on to the clinical trials phase. In that entire process, a process which will typically take anywhere between 10 to 12 to even 15 years, starting from about 10,000 compounds, you finally end up with one. Right? From the 10,000 odd compounds that you start with, just about 5 actually go into clinical trial. And clinical trials, you can see there are phase 1, phase 2, and phase 3. I'm sure you already know what these are. So phase 1 trials, typically looked at the safety aspects and the dosages, fixing up the dosages. The phase 2, and this is essentially for healthy volunteers. So you are really trying to find out whether the drug is safe to be administered. So in phase 2 is really where you are applying it to a target population which has a disease that you're trying to cure, right? So it really looks at the efficacy of the drug, right? And you typically have anywhere between 100 to 300 volunteers for it. Now, this is a phase which typically takes a long period of time because for most diseases, you may not necessarily have a big enough pool of volunteers who have the disease. So the recruitment of the volunteers takes some amount of time. Then you move on to phase 3 studies where you look at various kinds of dosages, you increase the population from 1,000 to 3,000. So you study various kinds of populations, right? Various demographic factors, all the time trying to see whether the drug is as effective as it is, and does it have any side effects? Is it safe enough? And this process typically takes 6 to 7 years. So this is the entire time frame it takes to bring a drug to the market. Now, it takes upwards of $2 billion, $2.5 billion is what it takes to bring a drug to the market. Why does it take such a long time? So if the previous slide, which took us through the drug development, discovery and development continuum, can be broken up into the stages, which you can see on the right here. So first, once you've identified the drug target, then you try to identify the hit. So that's a target to hit. Then there is a hit to lead. Lead would essentially be the drug which has all of the properties that you really want, right? Then you go on to optimize the lead to try and make sure that all of the characteristics, are optimal. Then is all the preclinical work which is associated with it. Then it goes into humans for animals, the safety and the efficacy studies. In the preclinical, you would have done, animal testing. Typically, you do at least two species tests. A lower species like mice, rodents would be used over there, and a higher species like primates or dogs, etc., would be used. Right. So if you actually look at it, you can see the first row is the most important one, right? So this is the probability of technical success. How you read it is, we said we started with 10,000 compounds. Out of the 10,000 compounds, 20% don't reach to the next level. Of the remaining, 75% only proceed to the next level. So another 25% drops off. Lead optimization, of the remaining, only 85% move to the next level. And when you go to preclinical, of the remaining, only 69% move on, right? So you can see a steady decrease. Now, when it comes to phase 1, and bear in mind that when you go to phase 1, literally you have kind of identified a drug, you established that it actually acts in the way that it is supposed to act. You have tested it in 2 species, right? Now, with all of those information, you are taking it into humans. In phase 1, 50% of the drugs fail. And this 50 is not from 10,000. It is, it has already dropped to this level. And from that, 50% fails in phase 1. And when it gets to phase 2, which is where you are literally trying it out in a person who has got the disease, only 34% moves to the next level. And from there, when it goes to phase 3, which is where you are doing the longer series of testing, 70% moves on. Right? So you can really see the issues associated with this and how difficult this entire process of identifying the drugs and making sure that, it actually works the way that it is intended to, how difficult that entire process is. Now, again, if you look here, now there's a specific reason why I'm, touching upon this slide and going a little bit more into the detail over here. Because when there is conversation around what does artificial intelligence, machine learning, automation, all of these do, that's where we will really come back to this, because cycle times, process improvements, all of those are essentially what you see over here, because you're looking at the cost, the cycle time, how much time it takes, right? Now, if you look over here, the golden colored portion, you will see that 17% of the cost is actually happening in the lead optimization phase. And that's the phase which involves a lot of human intervention, because this is you're finding better and better ways to optimize whatever that you have got, right? And that's also the space where there is maximum amount of money which is being spent in the preclinical phase, right? You can see the proportion of the money which is actually being spent over there. 17% is actually coming there, right? So when you look at artificial intelligence or any of those kind of techniques and tools to improve the cycle times, which is essentially means to bring a drug to the market that much more faster, or to reduce the cost, this is the area that you typically operate in. Now, when it comes to artificial intelligence, typically, we are looking at 3 aspects of it. I know if you read on the net, you will find literally, hundreds and thousands of links. For you to make really a sense of where does AI fit in all of this, you have to look at it from 3 perspectives. Typically, there are 3 things which the entire spectrum of tools which forms with, which is part of the AI landscape, help scientists with. The first one is automation, which is essentially all of the repetitive tasks. The AI system, AI automation, robotics, all of that helps to do. This includes all of the tasks. When tasks are automated, one of the good things which happens is that human error is eliminated. It also, by using robotics, it also allows you to, for example, pipetting, right? So you ensure that the exact amounts are automatically kind of, used. It also allows you to do relatively more dangerous, experiments, etc., without a human being being put in the face of danger. So there's a whole lot of things automation helps you to do. It automates a lot of the manual labor. It helps in ensuring that everything is tracked properly. Electronic lab notebooks, all of those digitization aspects are also part of the overall automation. The second thing that this AI system helps you to do is to spot patterns. This includes going through literature to find if you're going after a target, is there a target dossier associated with that? What is the existing literature on that? As well as based on previous experience, spotting things and trending patterns in the results that you're getting and helping you to analyze the results. The next part is prediction, which is essentially about looking at previous things and then predicting an endpoint, predicting whether something is likely to be toxic or not, predicting the stability of something, predicting whether the absorbs how a drug is absorbed. Is it going to be within our estimated levels or not? So these are really the 3 areas where AI will help you: automating things, spotting patterns and helping you to analyze things, and finally predicting outcomes. Right now, so when you look at all of this and you go back to this, you can see where artificial intelligence can help, right, in all of these kind of aspects. Now, here, one really, really critical thing is if you really go to phase 2 and look at the percentage of technical success, which is actually 34%, right? 1/3 of the drugs which reaches that phase, only 1/3 actually goes through. Now, one of the reasons behind that is that we just don't understand biology all that well. We really don't understand targets. Even though we have a fair amount of understanding now, we still don't fully understand targets. Without understanding targets, it is very, very difficult to create drug modalities and predict what exactly is the kind of impact it will have. Those would be areas where artificial intelligence could help us in the future, as well as a lot of these areas where speeding up things, predicting things, identifying patterns. And this is really where the entire spectrum of AI, machine learning, robotics, automation, all of that fits in. Typically, when you look at the discovery space, in the drug discovery space, there are 300 plus companies. I think right now it is much, much more. Probably 500, 600 companies are there in the world who are focusing entirely on the pharma R&D and AI. Okay, so typically, if you plot them on the X and Y axis, they come with either it's always a combination of expertise in drug discovery or expertise in AI. Now, I'm including this list, and there is also the sources also given at the bottom. You're free to, this report is available on the net. You can easily go in and check for it. Now, I'm showing this here so that it helps you understand where exactly do AI companies focus their attention on. Are they going for the full breadth, like an end-to-end AI platform, right? Or are they going for niche areas? And across the expertise in drug discovery, are you looking at preclinical? Are you looking at clinical? Are you looking at validated R&D use cases? So this sequence, where exactly are these companies focusing on? Obviously, it is impossible to have a company which does everything equally well, right? And one of the things that you would, in the last week, there was this, information in the market about IBM Watson, no longer being what they thought it would achieve. Somehow they were not really able to achieve it. That's actually one of the reasons is, where you are focusing, what are the problems that you are trying to address. Unless you define that very, very clearly, AI will not really help us. Okay, moving forward. Now, I've spent a lot of time talking about how difficult it is for drugs to come out into the market. 10 years, 12 years, humongous amount of money, effort, etc. Naturally, one of the questions that might be there in your minds is, how did COVID vaccines get developed in less than a year? So in this slide, if you look at, on the top, I've actually plotted the entire journey from target to hit all the way till submission. And this is the 10, 12-year journey that we were talking about. Below that are the milestones associated with COVID. Okay. Now, rather than reading through it, let me just tell you a few places where things really got sped up as far as COVID was concerned. So the first and foremost thing is, this was a crisis which affected everybody on earth. So there were unprecedented levels of cooperation. There was an amazing level of funding available, right? Within a very, very short span of time, December 1st is when it was documented in China. By 10th of Jan, the entire viral sequence, right, the genome sequence was published and it was available to people. In 14 days' time, Moderna was actually able to come out with their version of mRNA vaccine. So the availability of information throughout, and the close collaboration between companies which typically used to compete with each other was one of the very, very important things. The second thing is, while we talk about things like mRNA vaccine, this was the first time that we had a proper commercial launch of mRNA vaccines. Even the AAV vaccines were relatively new. But these were all technologies which had been in existence for a very long period of time, 30 years plus. mRNA technology took almost 30 years to mature. Similarly for, some of the other aspects, which we'll see in the next slide, right? The second thing is, I had mentioned that one of the areas where typically things slow down is in phase 2. So if you just go back, you will see here, 6 to 7 years is typically it takes in the clinical trials phase. Now, when it came to COVID, one of the things is that in phase 2, where typically you have I think somewhere around 100 odd patients, right? You need patients in phase 2. Because of COVID, because of the fact that there were so many COVID patients, you had no dearth of volunteers. And usually phase 2 is a very bureaucratic process because everything is documented. Okay, it goes through, approval agencies take their own time, recruiting of people takes its own time. Here, within very, very short spans of time, companies like Moderna, Pfizer, AstraZeneca, each one was able to get more than 30,000 volunteers. Okay, compare that against the 200, 300, volunteers, and the up to 1,000 volunteers, typically which are there in phases 2 and 3. Right, so that much, the presence of volunteers, speeding up of the phase, the clinical, trials, the everybody working together, right, and the availability of funding, all of these things contributed quite a bit. And finally, SARS had already come before. And in that entire family of COVID viruses, there were several instances, both in humans as well as in animals, which had happened earlier. Even this is SARS CoV-2, SARS CoV-1 was also there. And already there was a lot of work, which was underway in terms of identifying the drug targets, as well as, coming out with medicines. So all of those elements really came in handy and helped to bring all of this, to speed up the entire process that much more faster. Right. The other interesting thing is, if you look at this on the left-hand side is the mRNA, all of the developments which happened in mRNA technology over the years, and on the right-hand side, it's the lipid nanoparticles. Okay, so LNP, or the lipids, is basically the coating in which the mRNA is packaged. Now, you would have heard of Pfizer vaccines having to be stored under sub-zero temperatures, very, very cold temperatures, and you need a cold chain to ensure that, the vaccine is not spoiled. That's really because of, the lipid nanoparticles. The technology took a longer time to develop, and it is extremely susceptible to temperature. But even this, there has been a huge amount of technological advances over the year. We just needed a crisis to bring a lot of technological work which had already happened over the past 50 years to literally come together and come together in the form of a modality. Right, so that's another interesting thing. I mean, there is nothing like a crisis to bring everybody together and figure out something which actually works. Okay, moving on. This is essentially talking about how has the drug discovery, the process evolved over the years. Ray Deshays, who is one of the, who is a research head at Amgen, looks at this as 4 waves. Okay, so wave 1 is fundamentally serendipitous discovery. So you have compounds which have a non-chemical structure, but you really don't know how it works. And the interesting example that they give is that of aspirin. Okay, when it was discovered, we didn't really know the mechanism of action. And to be honest, we still don't know how aspirin works. Of course, people know that, okay, there is a COX-2 inhibition, but again, the exact mechanism of action is still unknown. I mean, if you search on the net, you will find several places where there are references to this. So the initial wave was all about understanding that, okay, certain, maybe plant products, etc., has a certain effect on the human beings. Can we produce drugs which will produce the same kind of a thing, even if we don't know what is it impacting? Then we moved on to rational drug design in the 1970s. So here we are essentially looking at specific compounds with known molecular targets that play a role in the disease. So, modulating a protein. So you know what the target is. Now you're trying to identify a structure which will bind with that protein and give us the intended effect. So I have a couple of interesting slides on that, which is there in the next one. So if you really look at structure-based drug design, let's use the example of finding a drug to treat COVID. Okay, so you know that there are already a couple of treatments available in the market. There is molnupiravir from Merck as well as Paxlovid from Pfizer. Okay, so this slide and the next slide essentially details out how Pfizer went about identifying an oral drug for COVID. So when you look at COVID, right, any kind of viral diseases, typically there are 5 areas where you can look at a treatment. One is you can look at the entry point itself, so entry or fusion inhibitors. The second one is inhibition of viral uncoating. So inside a host cell, the virus comes inside the host cell, then replicates. So you're trying to figure out a way of actually stopping this process. And there are five areas where you can actually develop a drug to do that. So entry, I mentioned, inhibition of viral uncoating, I mentioned. Then you can look at nucleoside and non-nucleoside, reverse transcriptase and inhibitors. Or you could look at protease inhibitors or inhibition of viral release. So these are the 5 areas. Now, as I mentioned, in the past, we already had SARS CoV-1. And at that time, several drugs were actually either in the final stages of being approved or pretty close enough to that. But by the time the disease kind of vanished on its own, so the approvals didn't come because we didn't have enough patients to complete the clinical trials. But there was already an existing amount of literature which pointed out the fact that protease inhibitors would probably be the best place to start. Now, what are protease inhibitors? So how a virus replicates is once it enters a host cell, it just gets the ribosome to create a copy of itself. So when the ribosome does that, you essentially end up with one huge polypeptide chain. Now, this is a non-functional chain. To make it functional, there are viral proteases, which essentially cuts this chain into its functional components and package it. So for the COVID virus to replicate and form another virus particle, which can actually inhibit, go and infect somebody else, it requires the protease enzyme to literally cut it into pieces. So cut it into, such that the spike proteins are formed, the nucleocapsids, the RBD, all of the various proteins and the other components associated with the virus. For that to be formed, the protease is required, right? So within the proteases inside the virus, inside COVID virus, the main protease or the MPRO is the one which was targeted in the earlier SARS CoV-1. So this again becomes a good starting point to understand more. And we already knew how the MPRO protease, right, the MPRO protease enzyme looks like. And they were also able to understand the 3-dimensional structure, identifying 2 locations where a drug could go and inhibit it. Okay, so these 2 locations were already known. All of this was already there in the literature, right? And as you, again, I've provided quite a few links here. You can feel free to go in and check some of these. Okay, now with all of this information already being there, when Pfizer started its process, they already had a working version of a drug, which was called PF00832-35231. These are internal codes which they use. By the time it comes out into the market, it will have a different name altogether. Okay, now these are all Pfizer drugs which you see on the left hand side. So nirmatrelvir is a technical name of the compound. Before it became nirmatrelvir, it was PF07321332. When it was released in the market, it started getting called Paxlovid. Okay, now this was the drug which they already had. Now there were 2 things. They were looking for a tablet. Now the reason for looking at a tablet is pretty simple. This PF drug was essentially an injection. Now the treatment for COVID, if it is an injection, it means going again to the hospitals, and producing injections are always again logistically more challenging, etc. So to do things in a much faster manner, you always needed an oral drug. a tablet, which can be, tablet or capsule, which can be administered orally. The problem with this drug is that it has got 4 NH groups. Now, this essentially means that if you swallow a tablet, it will not, get to the intended, region of action, because it will just not get past the stomach. Okay, so which meant that they had to do something with this, change this structure somehow, such that they get a final end product which is as active on the M-Pro as this particular drug is, the one in the middle. So if you look at the right hand side, you will see this is if this is M-Pro, it has got a SH group. Okay, so essentially what you're trying to do is to form either a kind of a covalent bond, or a bond with this, such that this is inhibited. That was basically what these folks were trying to do. That process meant that you had to figure out how to convert this into either this or this. So ideally, till the last stage, they had two candidates, okay, which were looking equally good. And this was the one which was finally chosen for a bunch of reasons. This was the one which was finally chosen. And the warhead within that is basically the nitro group. So you can see a nitrogen N here, right, which is the one which is actually going and forming a bond over here, and covalently inhibiting this. Now, in order to get to this structure, you can see some of the partial steps where you are actually, if you look at the molecule, the left-hand side, the middle portion, the right-hand side, each of those groups, how difficult it is to actually create something, synthesize this in the lab, and actually check whether it is working or not. Okay, and it also just shows how effective a company like Pfizer is, and how everything works so seamlessly and so fast. If for an R&D setup in the top pharma companies, this is the way in which they would work. But even then, you can see the amount of work which was required to kind of finally reach this stage. Okay, now all of the things which I've mentioned on this slide are actually there in the links at the bottom. Please feel free to kind of just go through them. If you are interested in medicinal chemistry, a blog which I would really suggest that you follow is something which is written by Derek Lowe. Okay, he has quite a few articles, and he writes it in science publication, science.org. You can please check him out. 2 articles over here are actually written by him. Now, moving on. So if you look at the waves of drug discovery, initially you had serendipitous discovery, then you move to rational drug design, which is what we just saw, how somebody kind of looks at the target that you are going after, and then you look at the structure which is required to inhibit that target. So in the 1980s, something interesting happened. That was the entire biotechnology revolution, which had recombinant DNA technology. That's where it started. Now you'll be hearing about monoclonal antibodies, biosimilars, a whole host of biotechnological drugs. Generally, we call all of them together as biologics. Fundamental difference between small molecules. So all of the molecules which are chemicals are called small molecules. The fundamental business difference here is that these are of biological origin. By biological origin, what we mean is that these are essentially peptides, right? So these are synthesized within either mammalian cells or microbial cells, and they are then injected into the human body. So to kind of understand a little bit better what are the differences between small molecules and large molecules. So the first point is, in the case of small molecules, it is chemical synthesis. In the case of biologics, it is generated by either mammalian cells or microbial cells. Size, it's called small molecules because typically there would be 20 to 100 atoms, and it is just about one kilodalton in molecular mass. Whereas in the case of biologics, it would be 5,000 to 50,000 atoms per molecule. Some would be even bigger. And usually, 1 to about 10 kilodalton would be the size, would be the molecular mass. Small molecules, when it comes to specificity, which is how exclusively it identifies a drug target and binds to it. Small molecules have much lesser specificity than biologics. Okay, so that's one of the reasons why there is a move towards biologics. All the bigger companies are really focusing on biologics. 7 out of the top 10 globally selling drugs today are biologics. Administration, this is another important aspect. Large molecules cannot be administered orally. They require systemic routes of administration. And when it comes to pharmacokinetics, large molecules has got a longer half-life, more limited volume of distribution, and longer time to reach peak concentration. So these are essentially the two differences between small molecules and large molecules. So one of the questions that typically you would look at when you are looking at a modality, these are called modalities. So when you look at a modality would be, once you've identified a drug target, you would look at whether it makes sense to do this as a small molecule or as a large molecule. Cost is much, much simpler for small molecules. Given a choice, you would always go for a tablet, which is the cheapest and the easiest way to produce a drug. But then depending on a variety of things, including all of the things that we have covered before, you would take a decision between small molecules and large molecules. Now let's do a little bit of a deep dive into this. So here you can see, the next two slides basically just looks at the state of the industry as on date and the various therapeutic areas. And you can see that, there is a shift happening slowly but surely towards biological drugs, biologics, and which can also be seen in the size. Now, if you look at the biologics which are out there in the market, and this looks at only the new new entities, right? So it doesn't include biosimilars. But if you look at this, you have monoclonal antibodies, which constitute more than half of all biologics which are available in the market, recombinant protein products, including humulin and all, cell and gene therapy, and of course the bioengineered vaccines. Now, here, just wanted to touch a little bit on the vaccines part of it, and specifically mRNA vaccines. Now, all of us are familiar with mRNA vaccines because of the vaccines which are released by both Pfizer and Moderna. Now, one of the really interesting things, if you look at the process part of it, is that you figure out the target pathogen, let's say COVID virus, you get the genome sequencing done. Once you've figured out the sequence, then you know exactly which protein to target, right? So spike protein is the one typically, which is targeted. Then the vaccine is designed and it is put inside the circle, which is this lipid nanoparticle that we spoke about. Then it is produced and then it is taken to the vaccination process. Now, the key thing to understand that is that if you are going for a different pathogen, if you are going for a new variant, the only change which happens in this entire process is a change in the sequence. Everything else remains the same. So here is where the modality in some sense is kind of becoming a process. So this is one very, very important change, and it's a very welcome change as well. And this also kind of portends to the importance of biologics and the newer technologies and modalities like mRNA vaccines and the entire mRNA technology itself, because it essentially means that the production times are that much more faster. Once we figured out the supply chain challenges, of course, we still don't have a fully functional ecosystem. Lipid nanoparticles, each of these technologies requires an ecosystem to develop around it, people who would supply all of the necessary materials. So that's still under development, but once that happens, the pace of all of these things will be much, much more faster. Just to give a sense, typically it takes anywhere between 30 to 40 years for any of these technologies to develop. So you can see the first mAb. mAb is a monoclonal antibody, came out in 1975. Now that to somewhere in 2026, we are saying that about 300 billion would be the total size of the industry for monoclonal antibodies. Now currently, cell and gene therapy or CGT is where mAb was in the 97-98 time period. If the previous slide where we talked about the breakup of the biotechnology sector, you would see that, the CGT, the percentage of their market share is pretty much lesser. But this is a space which is kind of, exploding like crazy. Let's just have a quick look at, overall cell and gene therapies and what really are they. So according to the American Society of Gene and Cell Therapy, gene therapy involves a transfer of genetic material, usually in a carrier or vector, and the uptake of the gene into the appropriate cells of the body. Cell therapy, on the other hand, involves the transfer of cells with the relevant function into the patient. Okay, let the definition stay there for a second. What we really mean, the fundamental difference between cell and gene therapy and any of the other therapies which came before it, small molecules, other biologics, etc., is that here you are literally trying to change something at the source. All of the other therapies were essentially treating symptoms. These were more symptomatic kind of a treatment. Here you are literally trying to change a gene, correct something. So gene therapy is fundamentally you are doing a gene addition, subtraction, deletion, inhibition, or editing, right? Some way or other, you are editing those, right? So here the technology is that you are, in some sense, it is called a living drug, because you are literally changing something at the source, right? And if it is successful, the drug stays inside the body. So in cellular therapies like CAR-T and all of that, the effect will be much longer lasting than in the case of traditional drugs, because once the the dose is stopped, immediately the action also stops. Whereas for cell and gene therapy, it is much more longer lasting. Now it is an extremely complex field, and there is a crossover between both cell and gene therapies. And because a lot of protocols, a lot of therapies actually use a combination of both gene therapy and cell therapy. So it is a very complex field. So the way to really try to understand it would be, what kind of gene modification strategy are you pursuing? So this is where you will come across terms like CRISPR-Cas, TALENs, zinc finger nuclease, etc. Then what is your therapy strategy? So here it is really about, how are you sequencing, how is your payload is basically what really does your work for you. How is it assembled? So mostly there are two varieties. There is autologous and allogenic. Autologous means that the treatment, whatever is required for the treatment, is derived from the individual patient himself or herself. Allogenic, it is not from any one patient. So if a patient has a disease, the cure for that, the cells, whatever is genetic material, whatever is being used, does not come from that individual patient. Unlike in autologous, where the everything is coming from the same patient itself. I'll show you an example as we move on. The next comes the packaging strategy. So packaging strategy is fundamentally about, now how do you get this inside the body to the intended location of action? So we would have non-viral vectors, right? The best example of that is the lipid nanoparticles. So the Pfizer vaccine, the mRNA vaccines essentially are all put inside lipid nanoparticles and put inside the body. The other packaging strategy is viral vectors. Adeno-associated virus is one example which all of you would be familiar with because of Covishield, right? So the AstraZeneca vaccine is an AAV vaccine. Even Sputnik is an AAV vaccine, right? And the Johnson & Johnson one is also an AAV vaccine. Then comes the delivery strategy. So is it an in vivo strategy, in vivo delivery, or an ex vivo delivery? So that also I'll just explain in one of the upcoming slides where I talk about CAR-T. It's much more easy to do it that way. So the next slide, as we go a little bit more deeper into this, I just want to spend a bit of time on one of the technologies which you would have heard quite a bit about, which is CRISPR, which is essentially a gene modification strategy. Now this discovery was what won Jennifer Doudna and Charpentier the Nobel Prize year before last. Now, here I've got a couple of book recommendations. One is called A Crack in Creation, which was authored by Jennifer Doudna himself, the Nobel Prize winner. The other is by Walter Isaacson, who has written an amazing biography about Jennifer Doudna and the whole CRISPR-Cas thing. Now here, these are some of the things which are genuinely science fiction like. So I'll spend 2-3 minutes just kind of talking about how this entire discovery came about, where is it coming from, why is it called CRISPR, etc. So so there are a kind of viruses called bacteriophages, which essentially attack bacteria. Okay, and this is a deadly virus. So it basically can wipe out bacteriophages, within a few seconds. Okay, its mode of action is that it basically just enters into the bacteria, replicates, bursts open the bacterial cell and goes out. So that's why it's called a bacteriophage. And on Earth, the number of bacteriophages outnumber bacteria. And the question is, how did bacteria survive? So one of the mechanisms that the bacteria evolved was something called CRISPR. CRISPR stands for clustered, regularly interspaced, palindromic repeats. Okay, so without really going into too much of the details of what those sentences mean, what those words mean. So basically what they, what the bacteria did was, it used a kind of an enzyme to cut off a certain portion from the bacteria while it was attacking. And then it got that portion of the genome embedded into its genome. And that portion was divided on either sides of the chipped off portion from the virus. They put up palindromes, right? Malayalam is a palindrome, you know that, right? So when you have palindromes, you actually know that what is coming in between the palindrome is the one which you need to kind of refer to. So in a way, the bacteria use this to identify phages when they are attacking. So that was a kind of a mechanism that they evolved to escape from virus. In a way, it is very similar to vaccination. So there are people who actually call it the vaccine card, which is used by bacteria to escape virus. Now, this technology essentially meant that there was a means of actually capturing a sequence, okay, which will help to identify something, and a kind of a molecular scissors, which will snip off this so that it can be incorporated. So the Cas part of CRISPR-Cas is essentially a molecular scissors, and the CRISPR is basically the portion that you want to change. So now what you see over the right-hand side is an example from a Kraken creation itself, where what they're doing is for a CLTA gene, okay, it's a gene associated with endocytosis. For a CLTA gene, the experiment that they did was they replaced a 20-letter segment. Okay, they replaced it with a different sequence. So for example, you can see a GNC which is replaced by an ANT here, right? Once repair and everything is done, this is the edited version. So this sequence, they once it was delivered to the point of action, the Cas enzyme cut it such that the original sequence can be snipped off and the new sequence can be integrated there, and it is joined, right? And they also added some fluorescence to it so that when you treat it, when you use a gel, you actually get a response, so you actually know that it has worked. So it's a kind of a, so you have a better and more detailed explanation here, but it is essentially a kind of a technology which allows you to very easily snip out a very specific portion of a gene and replace it with a corrected version. So if you have diseases, genetic diseases, where which are caused by a mutation, where some part of the sequence has gone wrong, you can actually replace it with this. Okay, still a lot of work is going on. There are companies like Caribou and all which are focused entirely on DNA, entirely on CRISPR, but the market applications are still evolving, but it's an amazing technology. Right, moving on to CAR-T. CAR-T stands for chimeric antigen receptor T lymphocyte. Now this is used in cancer. in cancer therapy. So if you look at it, you can see exactly what is being done. So in the clinic, you actually take the blood of the patient, then you take it to the lab. In the lab, the T cells, the T lymphocyte cells, are engineered to find and kill the cancer cell. This is by an inactive virus is used to insert the genes into the T cell. So finally, essentially what happens is these genes cause the T cells to make special receptors called CARs on their surfaces. Okay. Now these receptors are then multiplied, and then the new blood is put back into the body. Right. So in the lab manufacturing facility, you actually create the CARs. Then in the clinic, the CAR T cells are put back into the patient's bloodstream. Okay, this is a process which can be, finished in almost a day's time, maybe even, much more faster. And there are companies in India which are already working in this space, including a company which is associated with my company. So this is again a space which is very, very, very interesting. Now here again, the whole process is that you're curing the disease by essentially putting a CAR on top of the T lymphocyte, which will enable the T lymphocyte to specifically recognize a cancer cell. So the CAR is specific to this cancer cell, so it will immediately understand this is the cancer cell and go and fight it out. Now, cancer spread most of the time because the body's defense mechanism is unable to identify a cell as a cancer cell. Right? So that's one way in which they are treating it. And it's a very, very fast treatment. Already it is, there are several examples of it already working, and it's still not fully hit mainstream. It is still a very, very costly process, but this is again a technology which we will be hearing about for a lot in the coming days. Now, within this itself also, there are two ways. There is the allogenic one and there is the autologous one. Now, in this particular case, this is autologous, which means that all of that is required is literally taken from the same patient. Then this becomes personal medicine. Now, if it was, not from the patient, but from a generic source, then that is called allogenic. So that's essentially the difference between the two therapies. Okay. Now moving on. So all of this fundamentally is moving on to something called precision medicine. Okay. So this is an interesting example which, created quite a lot of flutter in Twitter last year. So this was the example of a 5-week-old boy who was admitted to the neonatal ICU. Within 37 hours, the entire genome was sequenced. The gene defect was identified. The treatment was ordered. It was received by the patient. Within 6 hours, the symptoms was resolved. So this is the kind of things, CAR-T and, some of these other newer therapies are promising. Still not within the reach of everybody. It is very, very costly. The ecosystem, the equipment, all of that is still not, reachable to the general public. And a lot of things still need to kind of, get resolved. But this is the future. This is where a lot of what we are, talking about is actually progressing. So now moving on. So if you really look at where we are so far, right? The three waves. The first one was we knew that something worked. Okay. But we didn't really know how it worked. Then we went on to, we knew what the drug target was. Now we are creating a structure which will selectively inhibit that. Now, the drug that we are producing, when you move to wave 3, was of biological origin. Wave 4. So all of this, all three waves, wave 1, 2 to 3, are one drug, one target kind of a model. In wave 4, you have something called multi-specific medicines. There are not too many examples of this, and there is one very, very interesting example which I will talk about in one of the upcoming slides. But these are essentially multi-targeted compounds or biologic medicines that mobilize biological activity to treat a particular kind of a disease. The main thing to keep in mind here is that this is one drug which will bind with 2 or more targets, which till now had never really happened. It is an extremely complex space. We are just trying to kind of, get a sense of it. So I'm not going to spend too much time explaining this slide at all. We have obligate multispecific drugs, non-obligate multispecific drugs. Some of these are very interesting kind of names, and these are names which you'll be hearing quite a lot in the coming years. PROTACs, which is there in matchmakers, is something which is already there in the market. There are several companies, which are already actively working in this space, in molecular glues, in tether bodies. A whole lot of this is already in place. The only difference between obligate multispecific drugs and non-obligate multispecific drugs is that in the case of non-obligate, it is not necessary that they reside in the same entity for the drug to function. So the multiple the binding activities, right? I mean, it doesn't need to be in the same drug for the action to actually happen. Now, I'll spend a little bit more time just talking about PROTACs, which hopefully will give you a sense of how all of this works, because otherwise it is a little bit too complex. Yeah, so PROTACs are essentially proteolysis targeting chimeras. Okay, chimera is a word you will hear quite a lot in a lot of the upcoming medicines and treatments. Now, this image which you see, and here I would really advise you, if you have access to this, to read up this article which appeared in I think it is Nature Reviews Drug Discovery. This is very recent. I think it came on last month. Here also there is one more by Ray Desais. Now these are people, Ray Desais and Cruz. Okay, Dr. Cruz. These are names which probably in the next 10 years would be winning a Nobel Prize. Similarly, Dr. Carl June for CAR-T. These are all upcoming therapies in very short span of time would become mainstream. Now, if you look at the image which you see over here, this essentially talks about how the multispecifics actually act. Okay. Now, okay, so it piggyback rights on a system which is already there within the human body. Okay, so there are drugs, there are proteins in the human body, sometimes which are not needed, okay, or has reached the end of life, or has not been created properly. Okay, it's misshaped or something like that. So those are proteins which needs to be degraded. Okay, because it doesn't serve any purpose. So the body has got a mechanism called proteasome to actually take care of it. That is what you see over here, this kind of a thing. Okay, it is fundamentally it is a protein shredder, like a paper shredder. That's exactly what it is. And you can see it is a kind of a cylindrical structure. It takes in the protein, okay, in its mouth, and it just shreds it into peptides. Now, for the body to identify proteins, it uses something called ubiquitin. So ubiquitin is almost like a post-it. So if a ubiquitin gets attached to a protein, then the proteasome knows that that protein has to be degraded. So it will just ingest it. You can see, one of those proteins in its mouth, right? And then just break it into its components. Now, what these scientists did was they figured out a way, and this is the PROTAC here, okay? They figured out a way to, once you know the protein which you need to take out, right? That is why it is called targeted protein degradation. Once you know the protein that you need to target, they figured out a way of clapping a ubiquitin on top of that protein. Okay, and that's why it is multispecific. So this PROTAC is, at binding to a ubiquitin as well as binding to a protein that it is targeting. And once that binding happens, this is how the structure looks like. Now there is ubiquitin on top of the protein that you want to degrade. So the proteasome recognizes that this has to be degraded. It ingests it and it breaks it down into peptide. Now the really wonderful thing is that this is a catalytic reaction, which essentially means that once this entire process is over, nothing happens to the PROTAC. It is free to continue this once more, which is completely different from all of the other kind of drug therapies that we have seen. Okay. Now this, there are several companies, including Arvinas, which is one of the leaders in the space, C4 Therapeutics. There are several companies. It's one of the most talked about therapies, these days. You can read it up, and definitely if you get a chance, read up this article. Now, why is this multi-specific? Because it is binding to two targets at the same time. Right? And this is used actually for degradation of something. There are several other ways. I mean, it's called X tags. I mean, it is light tags, ribo tags. There are several flavors of this. It is essentially used to kind of create, the impact by essentially binding to two targets at the same time. What this also means is, one of the reasons, for a lot of drugs to fail earlier, we said was that, there are only a very limited number of drugs which can be targeted. A lot of drugs, including what is called as KRAS, which is very, very important in cancer therapy, are undruggable. If you use this methodology, multi-specifics, what it also does is it induces proximity. So earlier, for a drug to act, it had to bind with a receptor site on a target. If you have something like a PROTAC, okay, or any of the multi-specific drugs, it will kind of, bring both these components together such that even if the drug is not binding with the target, it induces a proximity for the drug to act. So that's the other interesting and important thing. So if you really look at the entire evolution of the drug discovery phase, we started off with one drug, one target. Okay, the first step, of course, was one drug, unknown target. Then you move to one drug, one target. Then you moved on to one biological origin drug, one target. And now you are moving on to one drug, multiple targets, and induced proximity. So this is roughly the entire spectrum of the kind of activities which is happening in the industry that you are deciding to join. If there is a time for anybody to join the life sciences industry, this is that time, right? So I've across all of these slides, I've just tried to give you a very, very broad overview of what drives research and development in the life sciences industry. What are some of the newer things which are happening? Some of these things are, when you first hear about it, it completely appears like science fiction. But what are some of these changes which are happening, as well as given you some links which hopefully will help you to kind of, do your own research and learn more about it. Now, when you do that, one of the issues which you are most likely to face is that, it is frightfully difficult to make sense of all of this. There are so many modalities. This language is just not helpful at all, right? I mean, if you read this, it would take minimum a PhD and then some more years to even understand any of these words. And all of these words has got a very, very specific meaning as well, right? Like when you looked at CAR T, what a CAR is and why it is called CAR T is actually explained within the name itself, right? So the problem is, because of the way in which the nomenclature is and the diffused nature of how everything is happening, it becomes really, really difficult for you to understand. So which is why a diagram like this will probably help you. So you have to start at the bottom. Everything in the life sciences industry is essentially a quest for precision or specificity. How can you make the drug as specific as possible? How can you make it as precise as possible? Precision is more about, an individual level therapy, right? One patient, customized to a specific patient. And specificity is more broad-based, but it is about, how specific can you make the target, right? So all of the waves in that we saw of drug discovery were all moving towards more and more specificity, right? So that's essentially, how you need to kind of look at it. Against that, the knowledge of biology is continuously increasing. So the number of targets that we know, what we know about it, that the volume of literature is constantly increasing. Newer technologies and models are constantly coming in, right? The AI models that we talked about, in silico, CAD, all of those kind of things are, coming up more and more. There are even business models, my industry, the CRO CDMO industry, which helps a lot of companies with these kind of business model, the venture funding, all of those things are constantly increasing. And as they radiate outside, more and more of that will come into this circle, the quest for precision and specificity. So this is probably something which will help you to understand a little bit better about how this industry is overall growing. Now, having said all of that, this is for the future. I guess at this point in time, you are more worried about what are the next steps. And again, from a job perspective. So this slide is really how a biopharma organization looks like. So again, the things which you saw on top, target identification and validation, hit identification, hit to lead optimization, IND enabling, IND phase 1, all of those. These are the clinical trials phase, right? This is the value chain we already saw. Any fairly large pharma company, all of this would be organized across 3 very distinct streams, which kind of interact with each other, but are completely different at the same time. So the first one is a biology or translation value stream, which essentially is concerned all about the biology. Is the drug actually going to act or not? Is basically what it is focused on. Then comes a chemistry development, formulation, clinical development phase, which is what would be the final shape of this drug? Is it going to be stable? What are its physical characteristics? How will it physically be? Is what it is. The first one is how will it act? Will it act? That is a biology piece. Chemistry piece is how will it actually be physically? The third one is will it be toxic or not? That's usually a separate stream, a completely different department altogether. So the entry point into any biopharma organization will be in one of these 3 broad streams. Simply by looking at, how some of my peers and all have, continued with their journeys in the biopharma industry, naturally, at least in places like India, one of the, after M-Pharm, of course, one of the interesting entry points is the formulation. One of the reasons, of course, is that India is a generic capital and there's a lot of requirement for formulation, but formulation is one place where people typically enter. Chemistry is another equally good space. All of biology is also available. All of the pharmacology, all of those experiments, all of that is there. Biotechnology kind of qualifications more point towards the biologics side of things. But everything else within this is usually within reach for anybody with a BPharm qualification, provided that they have an appropriate higher level qualification. Safety assessment is also available, but this is also a space where a veterinary degree is of a higher level of importance because there is a lot of animal testing involved. Okay. So yeah, so that is more or less, what I kind of, wanted to share. I'm just looking at the time. I think it's been quite, I intended to close this in about 45 minutes, but it's been, I think a little bit over an hour and 15 minutes. In summary, if I were to kind of, talk about how everything is developing, 3 slides more or less captures everything. This is how the entire drug discovery development space is evolving. These are the new trends. These are the things which are happening around you. There is, of course, it is, joined at the hip with the efforts to optimize things, make things, work a lot more, make sure that the attrition rate in drug discovery is much, much more lesser. This is ideally the best way for you to understand how everything is evolving around you. And this is the structure of a typical biopharma organization. So I'll stop with this. Since I couldn't see the chat window, if there are any questions, Manoj, if you can help. If I can answer that, I can, I will answer that. Like I said, I'm not a scientist. I'm looking at it purely from how the industry is progressing.
Thank you, Harry. It was a very enlightening topic, and I have covered the status of the biopharma and in general drug discovery update, and it's quite knowledge heavy, and I think you need to go through that multiple times or maybe specifics to understand. But it's a very good overview of the whole research sector, and hope this gives the viewers an idea of where the industry is going. So there are a couple of questions that I can read it out to you. And one of the earlier question was regarding how does the multi-specific drugs induce proximity? I think you have answered that in one of the slide. Is there anything more to add? Otherwise, we can move on to next question on that.
Not much more. I mean, see, this is basically what it is. It is a space which is again, evolving very, very fast. Again, the article that I referred to, right? I mean, it's just hardly a month old. So fundamentally, the idea is that it brings both the drug and the target together. And even though it is not bound in the conventional sense, it actually induces a proximity between the two of them, which is good enough for the drug to act.
So, okay, basically, if I understand it correctly, it brings the payload close to the target by using this as a glue to connect between.
Yeah. So, which is why one of those terminologies is molecular glue. I mean, there are, of course, multiple variants of this, but yeah, that's essentially what it does.
Okay, that's good. There is one question on benefits of PROTACs over traditional inhibitors. Do you have something to know about that?
Again, it is evolving quite a bit, but again, the induced proximity is, of course, one of the most important aspects of this. And it's not just PROTACs; it's that entire multi-specific space, right? The ability to have a drug which will connect with two targets at the same time, right?
Okay.
And also the induced proximity, which essentially means that things which were undruggable in the past now start becoming druggable. So I will give a philosophical answer to this. So fundamentally, it is that it broadens your imagination. It actually allows you to pursue things which were, not considered possible in the past. Like the example which you see on the screen, right? If just imagine somebody who can dream up something like this. This is not conventional, right? It is coming completely, it is completely out of syllabus.
Definitely. So that is exploring more avenues into drug discovery and all of the biology options, what can be used to target a specific thing.
Targeting things which were earlier considered undruggable.
Undruggable. Okay, that's good. There's one question on virophages like Sputnik. There is a virus. This is not connected to the vaccine anyway. Virophages to use against coronavirus. Is there anything known about that?
So that's more of a scientific question. So I'll skip.
Yeah. Okay, sure. So what is the scope of therapeutic cloning in modern pharmaceutical area?
Okay, that's again quite a bit. Again, it will take quite a lot of time to kind of cover that. So I don't know whether it started off with the monoclonal antibody or any of those kind of areas, but see, rather than really going into those areas, I would say that use something like this to get a sense of where exactly the industry is at. Because even if you were to get a chance to get into something like this, I mean, you just have a broad understanding of where the industry is headed. Right? So unless you have a really, you really want to do something in a certain space, it's very difficult to get an area of your choice. This finally, when it comes to entering an organization, these are the streams where you kind of hit. So going too deep into something will not really help you much unless there is a very specific reason why you are kind of asking that.
Correct. There's one question on the Covishield excipients. It's called histidine and histidine monohydrate, and that is causing allergies. That's more of a specific scientific questions. Maybe we'll find answers and relate back to them. So I think that is largely the questions. Maybe I will add a couple of them from the students' interest point of view. Like we have talked about biologics and other aspects. So where do the conventional QSAR docking kind of studies stands now? Like I think from the curriculum point of view, I think this is what students are mostly studying in BPharma and MPharma. So what is the role of these activities right now in the industry?
It is very much there. It is already incorporated into rational drug design.
Okay.
Right. So all of the structure, activity, relationship. So once you understand the structure, right, of the target that you are going after and the drug that you are developing, that actually allows you to visualize what is the relationship between the two, and how exactly to come up with a drug which you like. So even in these examples which we talked about, right, all of those things are already inbuilt into that. Everything in rational drug design has those elements inbuilt into it. So when you're moving from in this slide, right, so all of those, I mean, QSAR is just one element of this. There are several other aspects as well. But so when you're moving from this, knowing that what you're trying to do is this, how do you decide on things, right? I mean, so that is where all of these studies actually come into the picture.
Okay, that's good.
This is also a place where AI and all kind of comes in into the picture as well, because there would already be examples of somebody else who would have done this.
Correct.
Right.
So I have added some more information on the group about AI, and if somebody is looking more into that, one key site to look for is the hardware providers. I will look on some links of NVIDIA.
Right.
So if you look at the NVIDIA GTC conferences, you will find a lot of companies using AI tools for various applications in drug discovery. There's one more question on side effect of booster vaccine. I think you may skip.
Yeah. So again, the reason why I'm skipping some of these questions, I mean, so I'm not a scientist. So one of the things which you will realize as you get more and more into this industry is that the more deeper that you go, the more difficult it becomes for you to get a sense of what is happening in the next room. So it is an industry where the amount of documentation, the amount of data, the amount of work which is required, and the amount of jargon which is there is just so much, right? So if you are trying to get an overview, then it is best to stick to that overview level, because if you go into the specific, it just kind of you better be an expert before you answer questions.
Okay. So one last question is about the specificity or the safety part of the drug. Why it is only accessed at the end? Why it is only visible only in the clinical trials? Is there any way to assess that upfront?
Yeah, so if you solve that, then you have you will be even richer than Bill Gates. Okay.
But I think there are some software to
no.
indicate a little bit.
I did mention this. Okay, so that question is basically this slide. Where are we? Yeah. So if you see, this is the place where it fails, right? I mean, 34% is 1/3 of the drug which reaches this phase. Only 1/3 actually proceeds to the next level. Okay, that is after 50% failing in phase 1. Okay. Now the point is, this is the first time the drug gets into a patient with the disease. Till it gets into the patient with the disease, we don't know whether it will act. In this entire space, we can do a whole lot of AI based design, projections, all of that. We can expand whatever we know on the basis of how it has acted in a mouse, how it has acted in a dog, all of those things. We are doing 2 species testing, but despite all of that, biology is much bigger than whatever we can comprehend.
Okay. So there's a good role of pharmacogenomics also in that.
All of those things come in. So eventually, this is a place where AI can play a role. It does the prediction. So some of these, I mean, to avoid, bad candidates reaching a certain space, yes. But these are rules of thumb. These are, there are still so much more we need to learn. We get better at it all the time, but we are nowhere near perfect in this.
Okay.
I hope I answered that question, because this is actually a question which I had as well, when I started on this.
Definitely. I think you had given some pointers for further exploration in that area. So maybe one last question, maybe for students' point of view, like in a career front. So what kind of qualifications are your colleagues or your scientists? How do they get into this biopharma industry? Maybe after BPharm?
Yeah, a
So,
lot of them have MPharm kind of specializations. Then they have done their, PhDs. So if I look at my organization itself, we have close to about 500 PhDs, okay, among the 4,000 plus scientists. And
okay.
over, I think about 125 or 140 of them, somewhere in that region, I don't remember the exact numbers now, did their studies in the US. And I'm mentioning that US or Europe or, outside India, basically. I'm mentioning that only because, I mean, there the industry exposure is a lot more richer because
Okay.
we don't do too much of original work in India. So it's not really the qualification per se. Of course, I mean, when it comes to specific areas, yes, those things are there, but it is fundamentally you need a PhD to really proceed further in this.
Okay. So after the pharma education, they focus on a specific area during their post
Yeah,
graduation
it
or
depends on where you
PhD.
want to go. And there are a lot of interesting courses, a lot of interesting, work which is being done in India also. The IITs, believe it or not, are one place where there's a lot of interesting work, especially in the cell and gene therapy area. IIT Mumbai, IIT Chennai, these are all pretty good. Indian Institute of Science. There are several Indian universities also where there are a lot of interesting courses which are more, connected towards some of the newer advances, specifically in the biotechnology field.
Okay, so that's great. So you have given an overview of all the new and upcoming research in biopharma, and anybody interested in this area can specialize on their area of interest and then get into the research domain, right?
Right. Within this also, I've not really showcased the ecosystem part of it. This is a typical pharma organization. There is, in some sense, it is also similar for the entire CRO, CDMO kind of industry as well. But there is a marketing role, there is a legal, there is the regulatory function, the vigilance function, there are project managers. All of those other roles are also there, all of which the same basic qualification and understanding of the industry is required, and a minimum MPharm is what I would suggest.
Okay. So there's one question about MTech in pharmaceutical technology versus MPharm. Is there any difference in that?
Fundamentally, there is not much of a difference. Uh, when we kind of had a discussion internally, I think the assessment was that typically it requires at least 4 to 6 months of training inside the company before a person can really be productive on the floor, because there is a lot of difference, a lot of things which are not fully there in the curriculum, and the hands-on exposure of a proper lab and actually doing the work, etc., it takes some amount of time. So the basic qualifications remain the same. There is still additional requirement in terms of training which needs to be given when somebody enters the industry. But from a pure entry point into company's perspective, I don't really think there's a difference between MTech and MPharma.
Okay. So I think we have covered most of the questions, and yeah, it's indeed a real good experience to review the new and interesting research domains. Thanks, Harry. Thanks for putting all this slide deck together, and it is a vast information. And I request participants to give a feedback on this, and in case anybody is interested in any specific area, because it's not possible to cover such a vast topic in one webinar, we can get specific expertise to explain you on each of that area. So request participants to give a specific feedback on what they are interested and what they would like to know more. With that, I thank Harry. Thanks for taking your time. I know you have a very busy schedule. Preparing the presentation and arranging that was not an easy task. So thank you once again. And hand it over to Ranjish.
Thanks a lot.
Okay, Harry, it was very nice. Needless to mention that it was very informative and exhaustive. I think the students need to put in more efforts to understand these sophisticated techniques in order for them to apply in their profession. Anyway, Dilip sir, are you there? Before Dilip sir? Okay, anyway, in the meantime, so as Manoj has mentioned earlier, we have completed this this edition of our webinar series. We have already planned for next webinar series. We'll be starting it soon. We have identified many experts and are ready with, they are all ready to share their experiences. Meanwhile, we request the students to share or mail their opinions or choices of their topic for the coming webinar series. Dilip sir, I think Dilip sir is not available right now.
Sir is there. Sir is there.
Sir is
Sir
there.
is there.
Okay. Okay. So I invite Dilip sir for to propose a vote of thanks.
Dilip, we are not able to hear you. I think please check the mic. So we're not able to hear you, please.
Anyway, Dilip sir, if it's not available, I will do the.
Sir, video off ചെയ്തിട്ട് ഓഡിയോയിൽ വരണുണ്ട്.
വരുന്നുണ്ടല്ലേ? Okay. Okay. Anyway, we will wait for him.
I think SAR is in somewhere bandwidth is not there, not correct.
Okay. Anyway, Prashant.
Okay.
Anyway.
Okay. So Ranjesh continues.
Okay.
SAR is coming and continue with that. Propose vote of thanks.
Sir, വരുന്നുണ്ടോ? Is he available?
The connection instability കാണിക്കണം.
Okay. Anyway, already Dilip sir had mentioned his inconvenience because I think he was traveling or he's not in his home. That's why.
Okay, fine.
Anyway, I take this opportunity to express our sincere, heartfelt thanks to Mr. Harry Jose, who has explained all the aspects of R&D in the recent trends in the R&D in pharmaceutical industry. On behalf of all the COPS global members, I express our sincere thanks to Harry Jose. And also, I take this opportunity to thank Mr. Manoj MN, who was the panel member, who has led this, who have been very successful in moderating the session. On behalf of all the participants here, I express our sincere thanks to Mr. Manoj MN. I also take this opportunity to thank Prashant SS and others who are behind this, working behind the screen relentlessly for the successful conduct of this webinar series. And also, I express our sincere thanks to all the students who have participated, who have keenly followed our series. And I take this opportunity also to thank our teachers. Chandran sir has just posted. Our sincere thanks to Chandran sir. So that's it from our side. Till we meet again. Bye-bye. Once again, happy Republic Day. Thank you.
Thank you.